Okay, so let's move to the next speaker. The last speaker is Dr. Harpreet Kaur. Would you please prepare? She holds the position of scientist at the Applied Sciences Team, Bioengineering Group of Plant and Food Research New Zealand. The title of her presentation is Can Aquaphotomics Improve Quality Prediction of Intact Fruit? Please start your presentation. Thank you.
You are mute, Harpreet. Harpreet, we don't hear you.
Mm-hmm.
Okay, now okay.
Kia ora, everyone. I am Harpreet from Plant and Food Research. Kia ora in New Zealand Maori language means hello. So today I'm going to talk about my PhD work that I completed last year on investigating aquaphotomics for fruit quality analysis. And basically, I want to thank my supervisors, Raina Kunamaya and Andrew McGlone, for giving me this exciting project on aquaphotomics, because I was aware of near-infrared spectroscopy, but aquaphotomics, when I started my PhD, it was pretty new to me. Thank you very much. And I would also like to congratulate Professor Senkova, and thanks to Professor Senkova for helping me, for guiding me, and Yelena for taking my questions, answering my questions when I sent you long emails. Thank you very much. So let me talk about my project. And basically, I'm going to start with fruit quality. So when you think of New Zealand, what comes to your mind? Beautiful beaches, beautiful scenery, or kiwi fruit? So New Zealand has got billion-dollar kiwi fruit industry. And basically, to have good quality fruit, we struggle very hard. We do very good quality research because consumers also demand high quality fruit. Talking about our horticulture export in 2019, we have the kiwi fruit, which has been exported to the world. Talking about the quality parameters, how will you judge a good quality fruit? It can be done externally, checked externally by color, shape, and effect of the fruit. And internally, everybody prefers good, sweet quality fruit. And we also check the dry matter of the fruit. The fruit can be measured destructively, which is done in the lab. And the two quality parameters, which are very important, are called as dry matter, which tells you how tasty your fruit is, and the soluble solid content of the fruit. The dry matter is basically done by taking a slice of the fruit. So you need to destruct the fruit, cut the fruit, and put it in an oven for 24 hours at a certain temperature, 65 degrees Celsius. So it's a destructive process, time consuming, whereas soluble solid is also destructive. You need to put juice of the fruit and take measurement. These two are very important because it tells you the maturity of the fruit and what's the right harvest time of the fruit. So we need non-destructive technique, and here comes light, near infrared light. So shine light on the fruit, take measurement, and it will tell you how sweet and what's the maturity of the fruit. So in this conference, we were talking about many portable handheld sun sensors. And in our lab also, we are using some of the sensors to find the maturity of the fruit. So on the left, we have Felix instrument F750, which is used to find the dry matter and sugar content of the fruit. We have also used the spectrometer, which works in the short wavelength near infrared region. We also have the DLP-NIR spectrometer, which works in the reflectance mode in the neospectra. So I will let you know how we are going to use aquaphotomics, so where the problem is coming from. So we make models, so dry matter measurements. This is the standard benchtop instrument, which was used to find out what's the dry matter accuracy. So error comes to 0.25%, which is close to the lab error of 0.20%. And Felix instrument, the one on this one, is also gave 0.28% of dry matter error, whereas SIO gave 0.38%. So we get very good prediction models, but they were done at one temperature. But there was a problem. We measure fruit in the second overtone region of water because the instrument, the light in the second overtone region has got high penetration depth. And this water peak, because fruit is 80% water, so this water peak basically shifts due to temperature. And when I have my calibration model at one temperature, for example, 20 degrees Celsius, and my model is going to predict a fruit at a different temperature. So what happens? I'm going to see an error. I'm going to see a bias. So my results are not accurate. And this is because of this wavelength shift due to temperature. This is the perturbation. So I was wondering, can aquaphotomics, because water light interaction can solve this problem? Whether we can have good prediction results using this aquaphotomics concept, or can we have a better hardware because of this aquaphotomics concept? When I started reading the literature, so most of the water bands were identified in the first overtone region of water, means all 12 water bands from C1, C2 up to C12 were in the first overtone region of water. But I work in the second overtone region of water for intact fruit quality measurement. So how I can use the aquaphotomics concept in the second overtone region of water. So that was the question, and how to solve this problem. So I started with simple experiments. I read the literature, so took Sucrose Solution. So in my lab, I had the Ph.D. NIR spectrometer that works in the 870 to 2500 nanometer range. And I took measurements just to see how different concentration of sugar. So this is the range of the Brix or soluble sugar value of the fruit. So how it is going to change. So the absorbance was decreasing with increase in concentration. So this is the first overtone region of water, which is in 1300 to 1600 nanometer range. On the other hand, I want to see the same thing in the second overtone region of water. So did measurement, and there was a similar trend. That's really good. So I identified the water matrix coordinate using the protocol defined by the aquaphotomics methodology. So water matrix coordinates were found. So different spectra was taken to find the water bands. Second derivative was used to find the water bands. Principal component analysis, PLS regression. So the water bands from C1 to C12 are clearly defined. So bands are clearly given. And I picked those wavelengths which were falling in these water band regions. So I was able to find the wavelengths, for example, 1414 here and in the different spectra. So that's the water band. But some of the bands were not in the region, but close to the water band. So this is all related to the first overtone region of water. How about second overtone region of water? How to find water bands, basically. So harmonic oscillator model or enharmonic oscillator model can be used. Harmonic oscillator model says the frequency or wavelength can be found and it should be the integral multiple of the fundamental frequency. So it's a symmetrical model and it doesn't take into account the harmonicity constant. But the molecule is not symmetrical because of the potential energy differences. So it's not practical, or we would say it's not the right approach to use harmonic oscillator model to find the correct frequencies. So I used both harmonic and anharmonic oscillator model just to calculate the frequencies. So these are already defined water bands, calculated water ranges, bands in the second overtone region using harmonic oscillator model, enharmonic oscillator model, and water bands. And the interesting thing was, when I made aquagrams, the enharmonic oscillator model frequencies gave me the same water spectral pattern as I achieved using first overtone region of water. So you can see the similarity between the two aquagrams. So with increase in the concentration of sucrose, the free water decreases and the bound water, these dimers, S4, S3, V1, V2, this combination strongly bounded water increases. And it's important to take into account the path length of the sample cell. You cannot use 1 millimeter path length cell to take your measurement in the second overtone region of water. You won't get appropriate absorbance or signal to noise ratio of the signal, a good signal, basically. So it means you're not going to get an interpretative aquagram in the second overtone region of water. Next, after using the sucrose solution, it's time to work with kiwifruit juice solution, because I cannot directly jump onto intact fruit measurement, because I need to wanted to know how I can apply aquaphotomics for aqueous solution, then going to solid samples. The measurements were done in both first overtone region of water and second overtone region of water. So you can see here in this graph, the measurements were done in the first overtone region of water from 1300 to 1600 nanometers at 3 different temperatures, 20 degrees Celsius, 25 and 30. And there was a peak shift, which is a horizontal peak shift, whereas for second overtone region of water, it's a lateral vertical shift. The isobastic point is 1444 nanometers, and here you can find it's after the peak wavelength of the graph. Now, how to find water bands which is related to temperature? So principal component analysis was used to find water bands. There's 1414 and 1494. So 1440 is the free water component, S0 species. And this one is the bound water species is 4, and corresponding water bands were identified and was able to get aquagram for 2 different overtone regions for kiwi fruit juice. But the question comes, how can I use this information to improve my prediction model capability? I use different chemometrics techniques, pre-processing techniques, for example, EPO. I also used EMSC technique. So what usually happens when we use EMSC or EPO technique? So the fruit or the sample is measured at different temperature to find out an interference matrix. But here you can see juice and water, they both have similar principal component loading coefficients, similar plot, same 1414 and 1494 water bands. And so instead of using juice as an interference component, I used water, independent water samples at different temperature and extracted the principal component and put it in the formula for EPO and EMSC just to remove the temperature correction, just to do temperature correction. And you can see the bias was reduced in both first overtone region of water and the second overtone region of water. So 0.39 to 0.09 in both first and second overtone of region. So use principal component of water. So water, molecule water, and water from juice, they were mimicking similar property when temperature was changed. That's a good thing. Another interesting application which was done in my lab with my colleague Mark Wong, he's working on chilling injury kiwi fruit. So he told me how aquaphotomics can be used to find chilling injury of the kiwi fruit. So what basically happens on chilling injury kiwi fruit? The kiwi fruit looks perfectly fine from outside, but from inside, because the fruit is stored at low temperature, 1 degree Celsius for gold kiwi fruit. So it looks perfectly fine, but from inside, there are water soaking tissues. So what the fruit is damaged from inside, but from outside, it looks perfectly fine. So how can we use aquaphotomics? How to get interpretation on chilling injury kiwi fruit? And in the first overtone region of water, and this is on intact fruit, the hydrogen bonded water increases in the chilling injury kiwi fruit. And for intact whole fruit analysis, so measurements was done using standard spectrometer, and I was able to find out water bands with low, medium, and high dry matter. So you can see the range from 14% dry matter to 21% dry matter. So free water decreases and bound water increases for high dry matter kiwi fruit. The experiment was also performed on apples, and it was also done for sugar quality analysis. And I was able to get good model for finding dry matter in 800 to 1000 nanometer region. And temperature correction can be done using intact whole fruit by measuring water at different temperatures. So use that principal component loading to do correction. So this was my PhD project, and what I have understood from investigating aquaphotomics is I got to know that aquaphotomics has so much potential to give us an insight about how free bound water species, and many more species can be found by using the methodology, and it gives us better understanding. For example, with temperature change, I got to know it's the free bound water species which is changing both in juice and water spectrum. And I can use the principal component analysis to do correction. So that's a wonderful thing I got to know, and we're writing papers to implement that technology. The sample path length is very important. Otherwise, you won't get meaningful aquagrams. So make sure you use accurate path length of the sample cell. So there was a trend for increasing the sugar content of kiwifruit juice and sucrose solution. So bound water was increasing, and there were different bound water species, S2, S3, and S4. And using principal component loading of water, so temperature correction can be done. And it was a very exciting project. And I really want to thank my supervisor. I want to thank Professor Rumiana. It was wonderful when I came to Japan in 2016 to attend the first Aquaphotomics conference of my life. And I got to know more about how Aquaphotomics is working. And after that, I started applying and implementing Aquaphotomics in my project. And in 2018, this is the picture, and I was able to give a speech, deliver a talk, and that was fascinating. And last year, I completed my PhD. So thank you very much. Thanks to everyone. Thanks to Yelena for inviting me. Thank you so much.
Thank you very much for a nice talk.
Thank you, Harpreet.
Okay, so the time has already come, and the schedule is already behind. So let's move to the next session. We want to close this session. Yeah, let's thanks again our speakers in this session.
Thank you. Can I Yelena?
Yes.
Can I suggest to start at 5 o'clock? So we are already quite tired. So
I'm okay, of course, 10-minute break, why not? I just want to say, because we ran out of time, this was such a beautiful presentation. Everyone who has questions, please feel free to put your questions in the chat box so everyone can see the comments, and Dr. Harpreet Kaur can answer to us. So just let's communicate, let's discuss.
Yeah, I can see that the chat works very well, because